the course Deep Learning by Example on Beowulf. This introductory course teaches the basics of deep learning and of different types of deep learning networks through a set of hands-on biological examples implemented in Keras, one example per class. Class number 6 will focus on graph convolutional networks, handling imbalanced data, and their application to classification of cancer types. This class is a part of a series, but each class in the series is supposed to be standalone. So for those who did not take previous classes, I will provide the background information necessary for understanding of the contents of this class. A few notes before we start. First, this class is being recorded. So if you don't want your face to be in the recording, please turn off your video. I will send out an email to everyone after the class when the recording is available on our website. Second, after each 50 minutes of the class, we'll make a short 5-minute break. So there will be 2 breaks during the entire class. And final, after presenting each slide, I will make a stop and suggest you to ask questions if you have any. Quick questions. If you have a quick question about the current slide or any previous slide, please unmute yourself and ask. Otherwise, please keep yourself muted. Let's postpone longer questions, statements, and discussions till the end of the lecture. The entire class is supposed to comprise the lecture, theoretical part, most of the class, and if we have time left, practical part. But typically, I know from previous experience, we may have from half an hour to maybe 15 minutes left for the practical part. However, all the practical part is available as a separate web page, a software manual, which should be self-explanatory. So you should be able to handle it yourself. But if you need assistance, please let me know. Any questions so far? No.
No.
I start. Yes, please unmute yourself, everyone, just to make sure. I'll start with the first slide now. So let's start with a question, posing the question. How do we represent and learn the structure of data? And the answer will generally depend on the type of data. In particular, in most of the classes prior to this one, we dealt with image data or sequence data, which are data with highly regular structure. Indeed, the image is basically a rectangular lattice of pixels, where each pixel has exactly 4 immediate neighbors. Or if you want to consider diagonal neighbors, it would be exactly 8 neighbors. And so the number of neighbors is fixed. And the sequence is one-dimensional lattice, where each element of a sequence has exactly 2 immediate neighbors. For these types of data, we used either convolutional or recurrent neural networks. And these both types of networks are specifically tuned for handling data with a regular structure. However, it's not always the case in practical applications that the data have a regular structure. Graph is a mathematical abstraction used to represent data with irregular structure. Graph comprises a set of nodes, or also known as vertices, connected by links, also known as edges. The node may be assigned a value or a set of values, which is termed feature or features. The number of links connecting a given node with other nodes is not constant. It may be 1, like here, maybe 2, maybe 3, maybe 4, or maybe even 0. So it may be any number. For handling this kind of data, we will use in this class the so-called graph convolutional networks. But we also will touch base on graph pooling layers, network layers. Although we have never dealt in the past with data that are described by graph, this is not something that we're totally unfamiliar with. Actually, in class number 3, we used gene expression data, for which we use dense layers, but in the principle, this data can be described by a graph, the so-called complete graph. In the complete graph, each node is connected to each other node. So that the degree of a node, which is the number of links connecting this node with adjacent nodes, will be constant again, but equal to n minus 1, where n is the total number of nodes in the graph. There is a number of practical applications where the use of graphs would be natural and would be beneficial. One popular example is social network, where the nodes are individuals, people, and edges represent connections between people. So these people communicate, they know each other. Another example is citation network, where the nodes are documents, for example, scientific articles, and the directed edge emanating from a given node to other nodes means that this given document cites other documents. The biological example to be discussed in this class is named GCN cancer, graph convolutional network cancer. In this example, nodes will be individual genes. Edges will represent some kind of association between the genes. And the data are similar to the data we discussed in class number 3. But the questions we ask about the data, or the tasks that we attempt to address, are different. And so accordingly, the approaches used in this class also would be different. I'm done with this slide. Any questions so far? No. Okay. Next slide will give a very quick overview of the main features of this class number 6, and as compared to other classes. What is the difference of this class from other classes? First, this class will take supervised machine learning approach, such as class number 1 and class number 2. In other words, we will have ground truth data with samples and labels. But the data, as I already mentioned, are very similar to data of class number 3. Second, this class will be actually the first of 2 classes to discuss graph neural networks. Therefore, I will not assume any previous knowledge about the graph, and the presentation will be given from ground up. So the purpose will be to demystify the graph neural networks. With this purpose in mind, the examples to be discussed in this class, all examples are kind of transition examples, in the sense that they can be handled by both graph convolutional networks and more traditional networks we discussed previously, such as multi-layer perceptron or dense networks comprising only dense layers. And they will be compared. The performances of these 2 models will be compared. And final, I think, important note just about why we consider graph at all. Why do we want graph for this data if they can be handled by other type of networks? The quick answer is that graph imposes certain constraints on the model and provides additional knowledge about the data. And that potentially would allow us to improve the accuracy of model predictions. And I will try to illustrate this point with a very simple toy example in the introductory part of the class, and example number 2, which will follow. Any questions about this slide? No. Proceed to the next slide. So we start with the first prototype or toy example now, which will give you the idea about the so-called graph classification task. This example will take as input synthetic ground truth data represented by this matrix, where the columns represent genes, the rows represent samples, and the values in the matrix are either 0, shown in white, which means gene is not expressed, or it may be 1, shown in color, red or green. The samples may be of 2 types. The normal samples, say normal samples shown in green color and assigned label 0, and the tumor samples shown in red color and assigned a label 1. Besides, within each type of sample, we assume presence of 2 modules of co-expressed genes, where the expression still may be only either 0 or 1, but with certain probability. And the probabilities of expression will be different across different models, but the same for all the genes within a given model. So you see light green, for example, it's module 0, dark green, module 1, and the same similar for red samples. And the purpose of this example is to train the graph convolutional network model on this data so that it could predict the class labels for new, previously unseen samples. Before we proceed to discussion of the model, let's get familiarity with some basic graph terminology. The terms shown here in green font will be discussed later in the class. The terms shown in red font will not be discussed further in the class. They are only presented for completeness of presentation as a possible alternative. So, as I mentioned, graph is a set of nodes connected by edges. For the models considered in this class, each nodes will represent individual genes. The edges will represent some kind of association between genes, and the features assigned to the nodes will be expression levels of the genes. All the edges of this graph, as you can see, are undirected. The directed edge is an alternative, corresponds to influence of one node of one gene on another gene. For example, one gene may regulate the expression level of another gene, but not vice versa. So we will not discuss, as I said, directed edges will not be discussed in the most of this presentation. As for some of the genes may not have nodes at all, not edges at all. These genes are termed singletons. In some cases, it makes sense to consider self-loops, the edge that emanates from a given node and returns to that node, or the multi-edges, when 2 nodes are linked by more than one edge or link. The multi-edges may correspond to multiple different kinds of associations between genes that are considered simultaneously. Indeed, later on in this class, we will discuss two different types of association between genes. However, only one type of association will be considered at a time. So multi-nodes will not be discussed. The graph shown here on the right-hand side represent the data shown in the table. Here, two nodes are connected by a link if and only if these genes corresponding to the nodes are members of the same module. For example, genes A and B are members of the same module, and both red module and green module. And nodes A and E are not linked because they are not members of the same model, neither in green samples nor in red samples. The graph shown on the right can be equivalently represented by the so-called adjacency matrix shown here, where in adjacency matrix, both columns and rows. Please unmute yourself. So the in the adjacency matrix, the value at the intersection of a given node and given a row and given column is 1 if and only if the two nodes are linked. Otherwise, it's a 0, and zeros are not shown here. This adjacency matrix possesses two important properties. One is symmetry. It is symmetric with respect to the main diagonal. The symmetry of the matrix is a direct consequence of the fact that there are no directed edges. If there was a directed edge in a graph, then there would not be symmetry. For example, edge from A to B would result only, this one would be 1 and this would be 0. And the symmetry of a matrix is an important property. We will take advantage of symmetry throughout the entire class. So the what I will present will not be applicable to the graphs with directed edges. The second important property of the adjacency matrix is that all its values are no more than 1. So there are no two values of 2 here. And this is a consequence of the fact that we do not consider multi-edges. If there would be 2 edges, say, some value would be 2 in the adjacency matrix. I am done with this class, with this slide. Any questions? Okay. If you have,
yeah, hi,
okay.
I have a question. Yeah, I'm a bit confused about the meaning of the modules. So are they assumed, and how do they relate to the data, the matrix that you assume as input?
Yes, they are related. As you can see, for example, first 3 genes are model 0, right? Module 0. Those are the genes ABC. If you see in green samples, they all light green or white, mean expression or non-expression. That means that all the genes in the module have equal probability of being expressed. It does not mean they all are expressed, but they have equal probability of being expressed. It's the same is true for the other model.
So the first 3 zeros correspond to A, B, and C, because I don't see the green that have always A, B, and C co-expressed.
Yes.
Is it probabilistic?
The co-expressed in this is highly simplified model. It's not real data. When real data would be different. I use this model for making some key points, some qualitative points, which we will see later on in real biological examples still hold. but this is highly simplified. The co-expression in this simplified model means that genes in the same module have equal probability of being expressed.
Okay, thank you. Makes sense.
But the probability between light green and dark green may be different. Or light red and dark red may be different. Any other questions? Okay, if you have questions later on, you can ask about including a question about this slide as well. I proceed to the next slide. Shown in this slide is arguably the simplest possible network capable of addressing the graph classification task. This recalled from previous classes that one way to graphically depict a network is to represent it as a set of data tensors, shown here as rectangles, and separated by layers or transformations between the tensors. This model includes only two layers with adjustable parameters: the GCN-conv, graph convolutional layer, and the dense layer. There may be other layers in the model, but they do not involve adjustable parameters, such as reshape, for example. There is some special flattened layer for reshaping the tensor, but it does not involve adjustable parameters. According to the convention we made in the previous classes, two layers with adjustable parameters is the minimum required in order for a network to be considered a deep network. So this network is deep. And we cannot simplify this network any further because the first layer is absolutely necessary. It constitutes the entire network. The first layer is kind of constituting layer. And the second layer is necessary for doing the classification task. Indeed, this with sigmoid activation function used by the dense layer, it will output a value, some number, between 0 and 1. And that number can be interpreted as the probability that the sample at the input of the network is tumor sample. Because if the probability is closer to 1, we will round it up to 1, and 1 is a label we assign to tumor samples. And if the probability is closer to 0, then we will round it up to 0, and 0 is a label assigned to normal samples. So that's about the dense layer. Now let's take a look at the graph convolutional layer, GCN-conv. What is significance of this layer? What it is doing? First important point: unlike any other layers we discussed in this course up to this class, this layer will take as input not a single item, but two different items. The first item is traditional. It's a sample, which in this case would be a vector of node features, a vector of dimension equal to the total number of nodes, and then so we a vector of dimension 5 with values node features, the expression levels. And the second item is unusual. It's adjacency matrix. And why do we need adjacency matrix here? The significance of adjacency matrix is that it defines the links present in the graph convolutional layer, because this is not dense layer. Not all of the links are present. For example, link from A to E or from B to E are missing. And that's exactly because there is no such links in the graph. From A to E or from B to E, links are missing. So the And there are some other number of missing links. So the present here only are the links that are present in the graph. And those links define the weights that are used in the formula for this layer. So for example, E value of element E would be weighted sum of elements of all the input elements, but except A and B, because they do not have link with E. For comparison, for the purpose of comparison, I would present also an alternative model, the multi-layer perceptron model, which is very similar to the model we discussed so far, with only exception that the graph convolutional layer is now replaced by dense layer. In the case of dense layer, there are all possible links present. The dense is also called fully connected. That means all the elements of the tensor X are connected to all the elements of tensor Y. And in this case, there is no need in the adjacency matrix, but theoretically, this dense layer could be regarded as a graph convolutional layer, but with adjacency matrix of ones. All the elements are ones, and that corresponds to the complete graph. And then the main conclusion from this slide: dense layer can be regarded as graph convolutional layer with adjacency matrix of ones. And by the way, the graph convolutional layer, which I discussed here, was first published by Kip van Welling, this publication, and then implemented in a software package Spectral, which I will mention later on in the class. Any questions? No questions. Okay, I'll proceed to the next slide. So we just have seen the role which is played by the graph in the graph convolutional network, which explains why the network is called graph convolutional, or layer is called graph convolutional. Now let's focus on the second part of the name, on see why it is termed graph convolutional. And for this purpose, I will compare the graph convolutional layer or transformation with more traditional, regular convolution, two-dimensional or one-dimensional convolution. I will discuss it on two-dimensional first, because it's easier to illustrate. So in two-dimensional convolution, we have a window of adjustable weights, or also known as a filter or as a kernel. And we slide this window over the input image X. And at each position of the window, we compute a weighted sum of pixel values and assign with some bias, and assign the result to appropriate pixel of the output image Y. And similar in one-dimensional convolution. This operation has, or transformation, has several hyperparameters. However, for the purposes of this presentation here, it would worth mentioning one of them, most important one, called padding. Padding characterizes the behavior of the filter when it approaches the edge of the input image. And Keras has two values for the padding parameter, the most typical use cases. The first value is termed same padding. The same padding is, we have this when the filter goes beyond the input image, and as a consequence, the output image has exactly the same size as the input image. This is why it's called same. Similar for one-dimensional case. The other case, valid padding. In the valid padding case, the filter does not go beyond the input image. As a consequence, the output image will have smaller size than the input. Now let's see what would be the analogs of the graph convolution or regular graph convolution with same pattern and with valid pattern in the case of graph convolution. I will start with the case of same pattern. The corresponding analog in case of graph convolution is termed filtering. So this operation, the transformation, is applied to input graph, shown in blue, and outputs gets the output. The output graph has exactly the same nodes. The number of nodes does not change. What is doing only reassigns the feature values to the nodes. And more specifically, for each node, say node A, the new value of feature will be computed as weighted sum of values of the input nodes, where summation is performed over the vicinity, or what is called one hop vicinity or one hop neighborhood of a given node. For example, for node A, that would be the one hop neighborhood would be node A, node B, and actually only A and B. C, because they are connected. A, B, and C are connected. So 3 nodes will contribute to this new value of node A. For comparison, the node B and D are not one-hop neighbors because they are not connected immediately by a single link. So they are 2-hop neighbors. By comparing this filtering transformation with regular convolution, we can see that the filtering performed using GCN-conv is an analog of a regular convolution with a filter of size 3. Why 3? Because when the size of a filter is 3, there are exactly we consider only the one-hop neighbors. If we would consider bigger filter, there would be 2-hop neighbors would be included, and so on. For example, filter may be size 5. So that's the point with filtering. Now let's see what would be an analog of regular convolution with valid pattern. In this case, we deal with not a single layer, but 2 different layers in the case of graph convolution. The first layer would be the same as we discussed, the filtering GCN-conv layer. And the second layer is so-called pooling layer. So the first I already discussed, but for pooling, in the pooling transformation layer, groups of closely related nodes will be identified and they will be merged into supernodes or child nodes, whereas the original nodes are termed parent nodes. And the child nodes would be assigned some feature values, also computed based on the features of the parent nodes. So it's called pooling or coarsening transformation. In conclusion, actually, I already mentioned that the filtering transformation discussed here is similar or analogous to the regular convolution with window of size 3. But later on in this class, we will discuss more advanced versions of the graph convolution of implementation, which may be regarded as an analog of regular convolution with kernel or filter of size more than 3. Any questions? Okay, looks like no questions. So I proceed to the next slide. If you have later on questions about this slide or previous slides, let me know. This code presents it's the simplest possible code that may be used to solve the graph classification task. It implements the network model shown in the previous slides. As we discussed in the previous classes, every code in Keras, at least the code discussed in this course, can be subdivided into 4 steps. The header, getting data, defining the model, and running the model. In this particular example, the header step includes a set of Python import statements, as well as setting some key hyperparameters that may be included, actually. Yes, key hyperparameters for used by the model. In particular, I mentioned here the GraphNet hyperparameter, which I introduced in this code. The code will handle both graph convolutional network and multi-layer perceptron. So depending on the value of this GraphNet parameter, we will see that later in the definition of the model. The second step is getting data. Here we define the models, as I already showed previously. We do some computation, do some looping to compute the elements of adjacency matrix. And we finally define the values X train and Y train. X train is the set of samples, matrix of samples, and Y is the labels, a vector of labels. The third step is defining a model. We use here the so-called functional API approach, where the intermediate data tensors used by the model are explicitly mentioned in the code. We define first, take compute inputs. There will be 2 inputs in this case. The input samples, as we discussed, and the second is adjacency matrix. Then, depending on the value of GraphNet hyperparameter, we either call the graph convolutional layer with a spectral library. The graph convolution is currently not a part of the standard Keras library. So we used here the library called spectral, which is also kind of written in Keras, but it is not part of official Keras. And if GraphNet is 0, then we just use dense layer for this model. And finally, the model will be complete. Definition of the model will be completed when we compile the model. Compiling the model means basically checking that everything what we need for running the model is in place and is defined correctly. In order for model to compile, we need to specify at least 2 inputs. One is the loss, and the other is optimizer. The loss is a loss function which will be minimized during the training procedure. Keras has a set of predefined loss functions, so it suffices only to specify here the name of the loss function. In this case, it will be BCE, which stands for binary cross entropy function, and optimizer would be, in this case, we take Adam. Also, there is a set of predefined optimizers defined in Keras, so we just specify the name of the optimizer. And the final step, running the model, we call a method fit, which performs automatic adjustments of the adjustable parameters in order to minimize the loss function. Any questions about this slide?
Yeah, I have a question. So here you generate the adjacency matrix, assuming you know the modules, right? And you use the modules to generate the data, the input data, right?
Right.
So in a real application, we'll have the input data, but we won't know a priori the modules. We don't. unless we use some assumptions, we wouldn't know what the modules are.
Mhm.
And so how do we provide an adjacency matrix
Oh,
for
that will be, of course, it will be explained when we proceed to real biological example, which will be the second part of this class. Of course, I will explain how we compute the adjacency matrix. It's somewhat different. This is very highly simplified example, but real example is somewhat different. But we will see a lot of similarities between the simple toy example and the real example.
Okay, thank you. And another question is that here, the way you define the adjacency matrix, because the modules are different for tumor and non-tumor samples, so you are assuming that uh, as long as two genes belong to the same module in either group, you connect them in the adjacency matrix, right? Because you have just one adjacency matrix for both tumors and non-tumors.
They actually we assume that they are connected, that two genes are connected, if they are members of the same module in at least one type of samples, either
Perfect.
in or both, of course, either in the tumor or a normal sample or both. Then we connect the genes in the graph, and we'll have one in the adjacency matrix.
Sounds good.
Otherwise, it will be zero.
Thank you.
You're welcome. Any other questions? Okay, let's proceed for now to the next example, next slide, and it actually will be a next example also, the second toy example. In the first toy example, our purpose was to introduce the graph classification task. Now we will take a look at the output, what this task is, compare the outputs between the graph convolutional network and the multi-layer perceptron, more traditional network. And the specific purpose I would like to look to what I have, and the specific point I want to make is about when the use of graph convolutional network may be beneficial, when we may have benefit in using graph convolutional network compared to more traditional networks. To this end, I will introduce, for this example, two new extra hyperparameters. So basically, it will be a very similar example to the previous one, but with two additional hyperparameters. The first hyperparameter is imbalance ratio. In the previous example, the probability of the sample to be green or red was the same, 0.5. In this example, we will assume that the data are imbalanced. For example, there are more green samples than red samples in the data. And the ratio of probabilities will be designated IR, for example, more green. Okay, so it's IR more than 1. The second hyperparameter is a number of singletons in the data. In the previous example, we did not discuss singletons, but we can now relax this assumption. We can assume that some number of genes are not connected to any other genes, F and G, for example, and in the corresponding adjacency matrix, all the rows and all columns corresponding to singletons will be identically 0. Now, I will try now to compare the accuracy produced by the graph convolutional network and multi-layer perceptron as a function of these two hyperparameters. But before I do that, I need to fix values of other hyperparameters that we mentioned already in the model, in particular the probabilities of expression within each module. I did not mention previously what are these probabilities, but let's assume that for light green, the probability is some value p. For dark green, was the second module, 1 minus p, and the same for red samples, p and 1 minus p. And I set the p-value to 0.7 for this example. Just we fix it and we don't vary it anymore. We only vary the imbalance ratio and the number of singletons. Although I showed in the chart number of singletons only 2, actually we use any number of singletons, 2, 10, 50, 100, 200. So rows correspond to different numbers of singletons. And the columns represent the results with different values of imbalance ratio. So if we see the chart shows number of correct prediction, percent of correct predictions from the two models. As you can see, with small number of singletons, the two models produce identical results. However, when the number of singletons is further increased, the numbers become different and they become consistently different. In particular, let's focus first on the case of balanced data. Imbalance ratio is 1. You can see that graph convolutional network consistently outperforms the multi-layer perceptron. You don't have to pay attention to specific numbers, but the most important conclusion is qualitative. We see that GCN works consistently better than the multi-layer perceptron. And I explain it as the following way. First, if data are balanced, the graph structure of data imposes a certain constraints on the data. For example, the singletons are not connected to all other genes. And singletons, to a first approximation, can be regarded as a noise. So in general, the more singletons we have, then the more noisy data are and the worse the predictions will be. And the way you can see that from the table. So the graph convolutional network does not include connections between singletons and other genes, and therefore the noise is attenuated in the graph convolutional network, whereas in the constraint agnostic multi-layer perceptron, everything is connected. All the genes, including singletons, are connected, and therefore it's kind of produces noise, contributes to the error of the prediction. Now let's see look at the case of imbalance ratio more than 1. I took only 10, for example. Here we see that opposite picture. At high values of singletons, the GCN, the MLP consistently outperforms the GCN. And I explained it this following way. The multi-layer perceptron has more adjustable parameters. There would be obviously some parameters corresponding to weights in connection of different genes. And therefore, it's more flexible in handling the challenge of data imbalance. So that's why it works better than graph convolutional network on imbalanced data. And we will see later on the biological example that many of these trends also hold for the real biological example. In particular, the singletons can indeed be regarded as a noise. With data, including singletons, we will generally get worse result, less accuracy. And when the data are imbalanced, okay, it's time for the first break. I will finish this slide and then we'll proceed from this point on. In the real data, we will see that when data are imbalanced, the analog of multi-layer perceptron, the dense network, we will discuss later, performs better than the graph convolutional network. That's it for now. Okay, any questions?
Gennady, there's a question from the chat. Is there any evidence that either of those models would behave better if you completely excluded singletons from the prediction task?
For this one, I think I showed. Yes, you can exclude singletons. You see behavior. Well, actually, the previous model is without singletons, so definitely you will get the answer. In general, you see at small numbers of singletons, there's no difference between the two. Okay, you see the smaller the number of singletons, the better the accuracy of predictions. The more correctly predicted, the higher the percent of correct predictions. So I think that addresses the question. If I can correctly understand the question.
So I have a follow-up question or comment. To me, it seems kind of an unfair comparison because the network assumes to know what the singletons are, right? Because you use that information to build your adjacency matrix. Whereas for the perceptron, you are not using that information. So if you were to use the GCN algorithm, building an adjacency matrix without assuming you know which are the singletons.
That's right. That's right.
Would they still outperform MLP when you have a large number of singletons?
The point is that if we make use of the graph structure of the data, then the model may have some benefit. With multi-layer perceptron, it is agnostic of singletons or of graph. It's just everything is connected to everything. Every gene is connected to every other gene. And you see that this results in less accurate predictions when the data are imbalanced. It's true that we are using the knowledge about what are the singletons, but only for graph convolutional network. For singletons, we don't know it at all.
Yeah, sorry, just a follow-up to my question. I was wondering if we completely removed the genes that are considered to be singletons completely from the model. That was my question. Is there any evidence that might improve either for the MLP or the GCN?
You suggest to consider multi-layer perceptron with singletons. Is that correct?
Without singletons.
Without singletons.
Yes, completely removed from the task.
Oh, I see. I see.
Yes.
Well,
so that every node will be connected.
Yeah. Well, actually, you should be able to do it with the first model easily. The first model does not include singletons, so you can because you will have the opportunity to run these simple examples yourself and change whatever you want to change. You can play with them. I don't have an answer right now available in the slides, but it should be very easy. Actually, the next slide will explain how you can run this models. The first model, you allocate interactive session, you load a module, and then you have two executables. One executable corresponds to the first example, and the other executable corresponds to the second example. So you have the files for this executable. You can download it yourself and change whatever you want to change, and then run it and see if you to address your question.
Thank you.
Actually, let's make we're already 5 minutes past the break. Let's make a break right now. It's 10:25, so we will return back at 10:30. Okay, it's now time to continue. We actually just finished the introductory part of the class, and we now proceed to discussion of the biological example. The biological example of this class is the application called GCN cancer, and it deals with classification of cancer types. So the purpose of this example to classify samples between a normal and 33 different cancer types. The data for this example are the same as in class number 3, and this is RNA-seq data that came from the Cancer Genome Atlas, or TCGA. TCGA is a NIH program led by NCI and NHGRI and aimed to identify a complete set of DNA changes in different types of cancer. In particular, the GCN cancer application takes 731 normal sample and more than 10,340 tumor samples of 33 different types. And the goal is to classify each sample as one of 34 different types. In addition to this TCGA data, this application also makes use of data from the STRIN database, which is STRIN database search tool for retrieval of interacting genes proteins. It's a database of protein-protein interaction. And the information from the database is used to determine the association between genes. In this case, it would be protein-protein coding genes. So this information will be used for computing the adjacency matrix for the case when the genes are protein coding genes. The application GCN cancer is now a module, a separate module installed on Beowulf, and it is re-implemented from the original version available on GitHub and published in this paper, and initially implemented in TensorFlow. The re-implemented application involves 4 executables, which should be executed exactly in the order shown. The first executable preprocess performs optionally balancing of training data. Optionally, it can perform custom adjacency matrices. The train.py executable performs training of the data on approximately training the model on approximately 80% of the ground truth data. Predict executable performs predictions of the labels for the remaining 20% of the data, and visualize produce some plots, normally bar plots of prediction error for different sample types. Any questions about this slide? Yeah. Produce the next slide. This slide actually gives initial overview of the code for the biological example. Shown here is the main function of the train.py executable. And there are many other files in the this application. This import options, data, models, all of these are separate files. So we import that information from those files, but the main function of the train executable is actually governs the execution of the entire code. And it, like we have seen for a simple example, it also comprises 4 steps: the header, defining a model, getting data, and running the model. The header involves the set of Python imports, including the spectral import from spectral library, the library of graph convolutional network functions. The defining a model, we actually will use 3 different models. The 3 different versions. One will include the GCN conv layer, the other Chebyshev convolution layer, which will be discussed, and the third will be extension of the multi-layer perceptron called dense networks. So only dense layers will be included in the model. Getting data, there are 4 different types of data that we will discuss later on. And we also will discuss how we can balance the data across different classes using the smooth variant library. And finally, running the model, just run fit method again, and it will produce the predictions, and we will be able to estimate the classification error. Any questions about this slide? No. Next slide. As I mentioned, the GCN cancer data involves samples from 34 different 34 different types, including normal type and 33 cancer types. This bar plot shows that this data are highly imbalanced in the sense that different classes, different types of samples are represented by a very different number of samples, ranging from the breast cancer represented by 1,100, more than 1,100 samples, to cholangiocarcinoma, just 36 different samples available. Furthermore, the normal samples shown here as orange bars are not available for all the 33 types of issues, but only for 23 types of issues. When we re-implemented all the data actually represented by 4 data sets, when this model was re-implemented in Keras, I have renamed this data set for convenience of usage. And the first 2 of the renamed data sets were, I assigned them the names GCE and GCES, where GCE stands for gene co-expression and S stands for singletons. So both these data sets are data sets with gene co-expression type of association. I mean, co-expression type of association between genes. And the adjacency matrix for this data was computed as follows. First, for all the genes in the data set, for example, for 3866 genes, we computed pairwise correlation coefficient, the Spearman correlation coefficients for between each couple of genes. And that gave us a huge matrix of 3866 by 3866, because there was this number of samples in the of genes in the original data. And these values of this matrix are all values between 0 and 1. Then we applied a fixed threshold 0.6 to produce the adjacency matrix from this weight matrix. So if the weight matrix of weights had a value more than 0.6, then the corresponding element in the adjacency matrix was set to 1. If the weight was below 0.6, then the value in the adjacency matrix was set to 0. That's about gene co-expression data. The other couple of data sets were named PPI and PPIS. And PPI stands for protein-protein interaction, and S stands again for singletons. So for this kind of data, we when computing the adjacency matrix, we used information from the STRIN database. It's a database that of protein-protein interactions. This database defines a confidence score for each couple of, well, by the way, this dataset includes only protein coding genes, not any genes, but only protein coding genes. And for each couple of genes, they define association score, which is a number, again, between 0 and 1. The score, the association may mean either direct physical interaction between corresponding proteins or participation of the proteins in the same biochemical reactions, as enzyme, for example. Or say proteins were, by doing literature search, by text mining, was found that a couple of proteins was often found in the same publication, which means also kind of association. They somehow associated. And for all of these cases, there are also in total 10 different criteria in the string database under which the couple of genes were regarded as associated. For all of these cases, the number confidence score was defined, which was a value between 0 and 1. And again, the threshold 0.6 was applied to this confidence scores. And if the confidence score was below 0.6, then the adjacency matrix was set to 1 for this element of adjacency matrix. Otherwise, it was, it was 0, otherwise it was 1. That's basically how we computed the adjacency matrix. Any questions about this slide? Okay, looks like everything's clear. Now I'll talk a little bit about the GCN cancer model, and basically what is the difference of this model from the models we discussed in the introductory part. First of all, this model classifies each sample as one of 34 different types, which means the output will no longer be a scalar. The output would be a vector of probabilities corresponding to different types of outcomes, and eventually we choose the sample type with the highest probability, and that would be final call. The second difference is that in addition to the GCN conv layer, we also consider Chebyshev convolution layer and dense layer as alternative. But Chebyshev convolution in this case is used as default because actually it works better on the biological example, on the data of the biological example than any other models, than any other layers. The third difference is balancing of the number of training samples across different classes using a specific technique, which will be described. And finally, optionally, there is support for doing pooling, for pooling layers. As I mentioned previously, you can merge some of the nodes into super nodes. And also, there is option for using multiple layers of graph convolution instead of just one. One is default, but you can repeat the same operation several times. Any questions about the model? Okay, next slide. Now let's take a closer look at the mathematical algorithm of the GCN conv layer. This is the simplest implementation of graph convolution, also known as vanilla graph convolution. And I will discuss these details using this very simple graph, the filtering of input graph X and output graph we have Y. And as we discussed previously, the new updated value of feature of a given node will be a weighted sum of values of the one-hop neighbors with some bias. The difference of this slide from previous one is that we now take into account self-loops. And that results in other words, when we consider one-hop neighbors of a given node, node A, for example, it will be one-hop neighbor of itself. So we include here the all the one-hop neighbors plus the node itself. And D here is the degree of the node. In other words, number of links of a given node to other nodes, including the node itself. So tilde stands for node for self-loop, for node itself. So this is basically the formula that is used for computations. However, this formula would be very inefficient to use directly. So there is an elegant mathematical formulation of the same formula. It's identical, but in matrix form, which would allow us to run this model on graph convolution on GPU, and will be dramatically speeded up. The computation will be efficient. First, how do we derive this formula? We first define a matrices, the matrix of nodes, and it's actually this is adjacency matrix, and this is degree matrix. The adjacency matrix, as usual, I already discussed that. The degree matrix shows how many nodes are connected to a given node. This degree matrix does not take into account self-loops. Then we define similar matrices including self-loops. To this end, we take identity matrix and add it to the degree matrix and to the adjacency matrix. We get now adjacency matrix, which takes into account the self-loops, and the degree matrix with self-loops. And then we compute the A cat, which is by multiplying the d tilde in the power minus 1/2, and then A and A tilde and D tilde minus 1/2. All these computations may be performed in Python easily, as shown in this code snippet, and the GCN-conf function is implemented as a part of the spectral library and can be called as shown in the slide. In conclusion, the GCN-conf is the simplest implementation of graph convolution. It's a local transformation involving the current node and its one-hop neighbors. Any questions about this slide?
What's the advantage of having the self loops, or what's the difference between in practical terms between including them or not?
I think that's pretty obvious. If you want to compute the updated value of, say, node, for example, node A, right? So you look at adjacent nodes, values of the adjacent nodes, and compute it as a weighted sum of features of adjacent nodes. So you regard the node A as a one-hop neighbor of itself. it will be different, obviously different number. I'm not sure whether it will be always better or worse, but its computation is different, because in one case you use only, I mean, in without self loops you would use only B as adjacent one hop neighbor, and with self loop you use both A and B in this summation.
Okay, thank you.
Any other questions? Okay, next slide. Now the vanilla graph convolution we discussed in the previous slide is the simplest possible implementation of graph convolution. In this case, in this slide, we will discuss somewhat more advanced implementation called Chebyshev convolution, and more sophisticated. And the reason why we want to consider this more sophisticated implementation is that actually it works better on our biological example. And the Chebyshev convolution takes a different approach. It's not local approach to computing the convolution. It is termed spectral approach. Spectral means that somehow Fourier transform will be used in this computation. And I will, to illustrate the idea of how this is done, I will first explain it on example of one-dimensional convolution of sequences in case of sequence data. It's a regular approach to convert. We discussed it already. We have a sequence, we have a filter or window W, and we slide the window and we compute at each position sum of pixel value, weighted sum of pixel values, and assign it to corresponding pixel, I mean not pixel, but element values, and assign the result to the corresponding element of the output sequence. This same procedure, which we looked at only graphically so far, can be written as a mathematical formula. You can think this is a formula. The W star X stands for convolution between W and X. This is star is just traditionally designation of convolution operation. This formula assumes that basically all the X, both X and Y and W are infinite sequences, but they are non-zero only at finite domain. So only colored blue elements of sequence X are non-zero. Others outside of this range are zero, identically zero. And you can compute it this way. There is, however, another approach to compute convolution for one-dimensional sequence, which is based on convolution theorem and based on using Fourier transform. According to the convolution theorem, a Fourier transform of a product of a convolution of two functions or two sequences equals a product of Fourier transforms of these individual sequences. So from this theorem, it immediately follows that convolution of W and X can be computed as follows. First, compute Fourier transform of W, then compute Fourier transform of X, then compute an inverse Fourier transform of the product, and you get the convolution. It's an alternative way of computing the convolution. This may look pretty cumbersome, but actually everything, all the computations can be performed very easily using fast Fourier transform. And this was first described in this paper. The fast Fourier transform, basically what it does is it decomposes every signal, every sequence, for example, as a sum of sines and cosines. And the output from the fast Fourier transform would be the coefficients, AK and BK. And why sines and cosines are used here? Basically, it's because these functions possess wonderful properties. They are form a so-called orthogonal basis of functions, which means any function can be decomposed in this way as sum of sines and cosines. And this wonderful property is a consequence of the fact that both sines and cosines are eigenfunctions of the so-called Laplacian operator, which in one-dimensional case is just a second derivative taken with negative sign. So indeed, from calculus, you can recall that if you take a second derivative of a sine or cosine, you will get the same sine or cosine, but multiplied by a certain negative number, which means sine and cosine are the eigenfunctions of the Laplacian operator. And that negative number, the multiplier, is taken with opposite sign because we have minus here, would be eigenvalue of the Laplacian operator. And all the eigenvalues of the Laplacian operator are always positive, except one, which is 0. And that one corresponds to value k equal to 0 here. So if the argument of sine, if argument is 0, sine is 0, but the argument of cosine cosine of argument 0 is not 0, it's constant. So if it's constant, cosine constant, we do second derivative, we get 0. That's why one of the values is always 0. So in summary, just this basically completes my very brief outline of the idea of using spectral approach for convolution of sequences. And now I'll illustrate how a similar approach, spectral approach, can be used for graph convolution. In the case of graph convolution, instead of the this values x, like x is a vector x1, x2, and so on, we now consider a vector x of node features of all the nodes of the graph. In this particular case, because we use highly simplified graph, it would be a vector of 4 elements. Instead of the Laplacian operator, we use graph Laplacian L, which is defined D minus A, where D is degree matrix and A is adjacency matrix. We defined them in the previous slide. Instead of the eigenfunctions of the Laplacian, we will use eigenvectors of the graph Laplacian, and eigenvectors are shown here in different colors. And you see eigenvalues are all positive except one, which is always 0. And there are also a number of wonderful properties possessed by the graph Laplacian. I have a separate slide which lists some of these wonderful properties. But let me now proceed to the final formula for the graph convolution. And by the way, all these computations can be performed also in Python, as shown in this code snippet. The final formula will be like this. Y, which would be the output from the graph convolution, can be represented as a sum with some positive k of this expression. Here, tk is a Chebyshev polynomial. The Chebyshev polynomial has a formula at some recursive computation. Chebyshev polynomial of degree 0 is just 1. The polynomial of degree 1 is x. I mean, argument x is the same as argument. And all Chebyshev polynomials of higher degree can be computed according to this recursive formula. But now, in this formula, instead of the argument x, we substitute the powers of the normalized graph Laplacian. The normalization means that we basically, like shown here, and the purpose of normalization is to make sure that all the eigenvalues of the normalized graph Laplacian are between 0 and minus 1 and plus 1. by using this formula, where lambda max is the maximum, the biggest value of the Laplacian, or graph Laplacian, which in this case, for example, would be 4. And the W in this formula is n by n matrix of adjustable weights. B is a also adjustable bias. And the final function, Chebyshev convolution, is defined in the spectral library like this. You can call it. The default value of K is 1. So K, again, is the highest degree of Chebyshev polynomial used in this formula. So unlike GCN-conv, the vanilla graph convolution, Chebyshev convolution takes a spectral approach, which is non-local by nature, because we actually, in computation of a given node, in filtering of a given node, we potentially may use all other nodes, not necessarily one-hop neighbors, just based on this formula. And also can be shown that when we use higher Chebyshev polynomial of higher degrees, that means that, as I mentioned, it will make use of the features of nodes, not necessarily in one-hop vicinity, but in k-hop vicinity of a given node. Any questions?
Hi, can you hear me?
Yes, I can.
I wonder if the in the red box, the lower red box, does the weight matrix is that a function of K, or is that constant for all the different Chebyshev coefficients?
No, W, you mean W, this function,
Yes,
correct?
W, yes. Sorry.
This is just a matrix. It's just a matrix of adjustable weights. Some of these weights will be identically 0. That will be defined by the term, this term. But otherwise, whatever, I mean, for some of the elements of the this W matrix, they will be identically 0 just because of the previous multiplier. But otherwise, it's not a function. It's just a adjustable weights.
Thank you.
Any other questions? Are you comfortable with this, or maybe what you are not comfortable with? I'll try to explain if something is missing. Anyway, this formula, even though it's pretty sophisticated and it's obviously very smart, it works better, as we will see from subsequent slides. And we have actually 2 important slides following this one. The first one is we will look at the classification error produced first using original or imbalanced data. This is, so we have in the first column is data type. We have 4 different data types: GCE, GCES, without singletons, with singletons, and again, PPI, PPIS. Then we have the percent of error for graph convolutional network. I call it GCN net. This is a network that uses the vanilla graph convolution layer. This is dense net. It's an extension of the multi-layer perceptron, but with output not necessarily a single scalar, but would be a vector values with the output. Then Chebyshev convolution. ChebNet is a network with Chebyshev convolution. For this case, we only present data for k equal 1 because this is the value used in the original publication. And we'll see interesting conclusions. First, dense net outperforms GCN net constantly for all values for all data sets, the dense net produces lower error than the GCN net. And that agrees with qualitative predictions of our prototype or toy model, where we have seen when data are imbalanced, the multi-layer perceptron performs better than the graph convolutional network. The second conclusion is that Chebyshev convolution, the model with Chebyshev convolution performs the best. It's best of all. It's slightly better than even than the dense network, even on unbalanced data. And it is also better than the implementation, original implementation reported in this paper. Although results are comparable, except for PPI dataset, where our implementation produces significantly lower error than their implementation. I'm not sure why that happened, but it's the case. And next, the third conclusion is that if we look at the error, the classification predicted error as a function of here different sample types, we see the most of the error, this one, actually corresponds to misclassification of two specific types of samples, the code and read, code colon adenocarcinoma and read rectum adenocarcinoma. Somehow it turns out that one is called instead of another, or vice versa. And I looked at the t-SNE in class number 3. We use, because we use a similar data, I had a t-SNE plot which visualizes all this data. It forms the clusters, joins the similar types of samples into clusters, and performs two-dimensional, produces two-dimensional plot of the clusters. And I analyzed this plot, and I found that the code and read basically have a mixed cluster. They are not easily separable, even using t-SNE, distributed stochastic neighbor embedding algorithm. And so I assume that probably in a way their expression levels are similar. There is some similarity between expression levels of these two types of cancer. So that's basically main conclusion from this slide. Any questions?
I have a question. So DenseNet doesn't use an adjacency matrix, right? Because it's just all the connections.
That's right.
So,
yes, it does not.
So the data type that you show here on the left column, those data types were used precisely to inform what those adjacency matrices are, right? Or what was that used for something else?
The adjacency matrix is used only by graph convolutional layer, either vanilla graph convolution or Chebyshev convolution. But in case of DenseNet, which uses only dense layers, there is no need in using adjacency matrix, because it assumes that it's fully connected layer. So everything is connected to everything. There is no any constraint, any limitation. So there is no need in adjacency matrix.
Right, but I understood that the data type GCE, GCES, PPI, PPIS was information, or were different methods for us to build the adjacency matrix to feed into the model. Is that correct?
No, no. Let me see it again. I'll return to the. Oh, actually, it's a few slides before. Okay.
I was just wondering why I was expecting the DenseNet to be the same regardless of what data type you were using, because I thought the data type
The
adjacency matrix.
Adjacency, the type GCE, GCES differs only in number, presence or absence of singletons. And singletons indeed are either included or not included in the data set. It's just a set of input samples. And these same samples are used by whether it's graph convolutional network or a dense network, they use the same set of samples. But the adjacency matrix is used only by graph convolutional layers, both graph convolutional Chebyshev and vanilla graph convolution, but not are used by a multi-layer perceptron. I mean, sorry, by dense network.
Thank you. Actually,
sorry.
I'm sorry.
Okay, yeah, just to follow up on that question, was there any instance in which the data matrix was the expression data, while the adjacency matrix was built off PPI?
The question was what?
Is there any instance, or is it feasible to use the gene expression data as the data matrix and the PPI as the adjacency matrix?
Oh, you mean whether it makes sense to use both? Correct?
Yeah.
Well, it probably is. I did not try because I tried to follow the original publication, and they do not. Actually, I used even these matrices that I used by default came from the original publication. So there is a way to generate your custom adjacency matrix. And my re-implemented version of this application allows you to do that. I played a little bit, but not too much, because there are too many different possibilities, too many different parameters. I spent limited amount of time, and I did not try to do actually both. But it's possible, it's doable. If you are interested, you can do it yourself.
Thank you.
Gennady, to clarify though, PPI still uses gene expression as the node values. It just uses strings to generate the adjacency matrix. Is that correct?
Oh, that's right. That's right. Both PPI and GCE data sets, or GCES and PPIS, they are used using gene expression samples. The samples are RNA-seq samples. Yes, but in PPI, we use only subset of genes, only protein coding genes, only protein coding genes, whereas in GCE, we use genes on any type, both RNA coding, protein coding, whatever is available in TCGA. Does this address the question? Okay, I'm returning to this slide, and now next slide. If there is no more questions about this one, I'll proceed to the next slide, which attempts to perform similar computations, but this time using the balanced data and using only the Chebyshev convolution. For this kind, I performed many computations, but this slide reports only the results for Chebyshev convolution layer network because it's performs the best out of all. So, and the main outcome from this slide would be that the balancing of data and using Chebyshev polynomial of high degree will dramatically reduce the classification error. And first, let's talk about balancing data. How do we balance data across different classes? I'll consider first example. There are two classes, for example. This is imbalanced data. There is majority class and minority class. And there are two fundamental approaches to balancing. One is called undersampling, where, okay, it looks like it's time for a second break. Let's make a break and then return exactly to this again, to this slide again. It's 11:15, so we will come back. Okay, it's time to start. There are two fundamental approaches to balancing the data. The first is termed undersampling, and the other is termed oversampling. In the undersampling approach, we remove a certain number of samples from the majority data set. And this may work, and there are some implementations of this approach. However, we lose data this way. So more preferable is oversampling, where we add some new samples to the minority data set. And there are a number of ways of doing it, how we generate new samples for minority data set. The simplest one is termed naive balancing, which just duplicates some of the samples of the minority data set, randomly selected samples, picked and duplicated. This does not work well, however. I tried myself also. It's kind of unstable. You have many identical samples in the data. If you train on this data using small batches, eventually some batches may include only identical samples, which kind of unstable. Result will not be, you will not see continuous decrease in the loss as a function of iteration. A more smart approach is using a special library called smooth variance, where smooth stands for synthetic minority oversampling techniques. That was first published in 2002. The idea was published in 2002, and 2019, a large number of different variants of this technique published by different authors were summarized in a package, which I use as a part of this application, smooth variance package. Currently it includes 92 different techniques. 2 years ago, when this class was taught for the first time, there was 85, now it's 92. And I played with many of these techniques. Some of them do not work, do not help. But I identified two techniques which work specifically well for the GCN, for TCGA data, using the models that we use. So if we balance the data across different classes using these techniques, I get much better results. For example, for Chebyshev convolution, with the same K value of 1, as we discussed in the previous slide, we have about 10-fold or more reduction in the error, classification error. You recall that in the previous slide, the error was about 5% or something, 4 to 5%. Now it's 0.9 for one case, and 0.25, this is 10-fold increase. This is even more than 10-fold decrease. And if we, however, use not Chebyshev convolution layer, not with K equal 1, as the same as was in the original publication, but if we further increase the degree of Chebyshev polynomial that is used by the layer, then it turns out that for GCE dataset, for example, we have 0 error for this particular data set for particular fold that we discussed. And for PPI, without singletons again, we have also dramatic reduction in the error, only one error per about 2,000 samples produced. And with singletons, the error will be higher, which is kind of agree qualitatively with what we observed in the prototype models. Singletons can be regarded as kind of noise, and when singletons are included in the data, the predictions will be noisy and would be less accurate. And the plot, similar to the plot that I showed in the previous slide, now look like this one. So there are 0 errors for many types of sample, and for some of them are very small. This plot is produced, by the way, for GCS data set. So in the previous slide, the same data set was used. So reduction is approximately 10-fold reduction of the total error, These data were balanced using MW mode algorithm, but LLE's mode similar. They're very similar output, so about the same result. And the conclusion from this slide is that the error in multi-class prediction from the TCGA data can be reduced dramatically, first by balancing the number of training samples across multiple classes. I mention specifically 2 smooth variants that worked well for me for this data, and the further improvement, further reduction in the error can be obtained by using Chebyshev ChebNet with Chebyshev polynomial of higher degree, for example, 20 or higher. Any questions?
Yeah, is there a performance implication of using a high degree of Chebyshev polynomial?
The question is whether it's popular application.
No. Is it computationally more expensive?
Oh no, not much more expensive. Not much more expensive as it's comparable, I would say. Maybe it's a little bit more, but comparable to other techniques, because it's actually a very efficient technique. You use graphical processing unit. There is a formula. No, it's not a big deal.
Thank you.
And actually, you can use your data for this. This code with your data. The only thing you need to do is to put them in the input format similar to the format used by this GCN cancer application.
All right, can I ask a question as well?
Sure.
So because of the way oversampling works, it makes assumptions of how the data is distributed, right? So is there any risk of causing overfitting on the types of training data that we input?
Overfitting, we would get overfitting when the amount of training data is very small and the model has a lot of adjustable parameters. In this case, by balancing, we actually increase the number of data. So unlikely, there is no chance actually that you can get overfitting by the oversampling technique. With undersampling, maybe, but oversampling, which I use here, no, there's no way you will get overfitting. You should be fine.
Okay, thank you.
And by the way, the new samples are generated according to these techniques, MW mode, or other techniques described in the smooth variance publication. This I generate completely new samples according to using some k-means clustering or something. Some techniques are described. Each variant is described in a separate publication. And so each technique is different, but it generates new samples, not just duplicates the samples like I mentioned in the naive balancing. Okay, if no questions, I just quickly overview the commands that you can run in order to reproduce this result yourself. The details are described in this user manual page. It's a separate model instead of installed on Beowulf, so we can load model GCN cancer, you allocate an interactive session with GPU, then load the model, and you have this. You can list the total number of source files. The source files used by this application. One of the source files is in R, used for visualizations, and others are Python. And you can run the executables, preprocess, train, predict, and visualize in the same order as I've shown. This is just examples of how you can do it. More details are in this user manual page. And finally, just a summary of what we have done in this class. It was two parts. It was introductory part where we introduced the graph classification task. We introduced the graph-related terminology. We discussed how graph convolutional layer is related to dense layer or to a regular one or two-dimensional convolution. We have discussed a second example, prototype example with imbalanced data, and we found that presence of singletons and data imbalance may reduce the classification accuracy. And we provided also some kind of insight into when the use of graph convolutional network may be beneficial because it imposes some constraints on the model, which helps to reduce the classification error. And in the second part, with biological example, we discussed two types of gene association, the gene co-expression type and protein-protein interaction type. We discussed the GCN cancer model, which has multiple options, and optionally can, by default, uses the Chebyshev convolution layer, but optionally can use GCN conv layer and dense layer. And we discussed the balancing techniques and the result, how this balancing and using Chebyshev polynomial, the Chebyshev convolution layer with a higher degree of Chebyshev polynomials allow reduce the classification error. That's it. Now it's time for more longer questions, statements, and discussions if you would like to make any.
Yeah, I have a question that is of a more general nature. So this works very well as kind of black box to predict, as you show in this example, to classify all these different cancer types. Is it possible to somehow use the weights that the model has learned to gain some degree of interpretation of on what's different between the different cancer types? So, like ideally, I would like to see, for instance, what are the gene networks characteristic for the different cancer types and how they differ from each other, or be able to compare them. Is it possible to somehow dig into the model and extract that type of information?
Actually, the model is trained on all the cancer data at the time. I mean, the way I did it so far. But optionally, you can do it yourself on a subset of, say, cancer type, even one type, say, use one of the cancer types and a normal type, say. You can train model on just subset of data. And you will get, of course, you will get different weights. So I'm thinking how you could, whether at all, and how you could use this customized training on specific subsets of TCGA to make discrimination between different cancer types. Actually, I'm not sure you need that because looks like the model works pretty well even with a single.
The question is, going parallel with any other machine learning technique, and even, I don't know, the simplest one, linear regression or whatever, or any classification tool. The goals might be to classify, but also another goal is to interpret, to understand what the difference is between the classes that you are trying to find. So in a very simple regression model or simple machine learning model, interpretation is an important component that you gain on top of the ability to classify, right?
Right.
So
Right.
here I know that, I mean, I don't know really much except what I learned from your classes. So I'm a total beginner in terms of using deep learning models. And I know that one of the criticisms is that it appears to work a lot more like a black box. So it's very good at allowing us to predict or classify, but it works as a black box, so it's very difficult to look inside. But I read or heard some about interpretability or I don't know, coupling these deep learning models with the ability to interpret what the models are, what they are doing under the hood, let's say. In this case, in terms of, to be more concrete, in this case, we are talking about gene networks. So definitely it would be quite interesting and illuminating to be able to interpret those biological networks, those gene networks working differently in different cancer types, for instance. Is that anything that one can do with these models to gain that type of information?
I think that, well, it's my impression, my opinion is that probably what you could do is to try to compute multiple different metrics. I'm using only one metric, the error, classification error. But there are many different metrics described in the literature. People now looks like actually starting last year, there is some kind of booming. This area starts to booming. I've seen before 2024, I saw on average about one paper per year published using this balancing techniques and using TCGA data. But starting 2024, there are 4 papers, and earlier this year already 2 published. So there are many different approaches and many different metrics used by the authors. Maybe the use of different metrics would help you to get some insight into what is actually the difference between the different cancer types. That's what I think I would try to do.
Okay, thank you.
And implementing this metrics probably also doable with our implementation, but I just did not do it because I was trying to follow the GCN cancer original application.
Can you share references?
Oh, yes. Well, actually, I can send you after the class, but if you go to Google Scholar, and just type TCGA SMOTE variants, or just TCGA and SMOTE, you will get a set of links. Most of them, as far as I could see, are starting from 2024. Looked like my previous class was taught in 2023. At that time, there was not so much as far as I could see, but now it looks like it's growing. The number of interest in using this balancing techniques for cancer data. They're kind of different application. Some publications discuss specific types of cancer, like only breast cancer, for example, and they compare whether or not breast cancer is present in the sample. Like this, it's different kind of task, different from what I presented in the class. But many of them, they present multiple metrics. I think that might be the way to get some insight, more insight into the reasons why you get the differences between different samples.
Thank you.
You're welcome. Email me if you would like assistance with, because I downloaded a number of papers. I can email you particular references if you send me email after the class. Any other questions?
I have another question that's very generic. So this could be applied, I don't know, to the perceptron, the simplest deep learning models that you discussed in previous classes. It's about the weights, the initial weights. Are the weights initially assigned randomly?
Yes.
Okay.
Yes. Well, and there is, if you look at Keras, at keras.io, and look for initialization, there are multiple different ways to initialize weights before you start actual training procedure. There is Glorot uniform, I think, the initialization method called Glorot uniform is default, I think, for Keras. But there are optional. You can, for example, set them to all zeros, starts from all zeros, if it works better for you. Of course, there is no way to predict which one will work better for your particular task. But yes, the answer is yes, you can use different ways of initialization. And typically, they are, by default, they are assigned randomly, but according to certain distribution.
Okay, thank you.
Otherwise, you can look, in addition to this class, to this presentation, you can take a look at the GCN Cancer user manual page to see how all possible options. You can see how you can generate these plots in particular, how you can train, how you can balance the data, some commands for pre-processing data, for example, pre-process -b, and then smooth variant MW mode will be, you take GCE as data set and then you balance it using this technique, for example. Or you can balance without technique, you use naive sampling balancing. And many other details about how you can use the software are available in this manual. I think it's self-explanatory, so there is no, you should be, I believe you should be able to handle, to figure out yourself. But send me email if you need assistance. Okay, we have 15 minutes left of the entire class. If there are no questions more, we can either finish, or if anyone would like to play with the software, we can continue and